A biological process is another one of these hard to define precisely terms. Examples include biosynthetic pathways, translation, transcription, replication, and secretion. The gene ontology folks define it as a recognized series of events or molecular functions, with a defined beginning and end. Let's examine this concept as it applies to a biosynthetic pathway through our state machine representation. Remember that we can model an enzyme in terms of a Mealy machine composed of the various microstates that the enzyme participates in. The transitions between these states can have inputs and outputs, and the scope of those inputs and outputs can be captured as constraints. We can model the constraints defining the transition between the ES and EP states by organic mechanistic constraints that define the molecular requirements for doing the chemical conversion. For example, a protease may recognize various molecules that look like this dipeptide, but many different R groups would also be good substrates, and presumably this amine on the far left of the molecule isn't necessary for the chemistry to happen. From an organic chemistry perspective, all that matters in this molecule for the hydrolysis reaction to occur is that the C double bond O-NH portion in the middle is present. That set of atoms represents the mechanistic constraints that all substrates must have to undergo this state conversion. The other two transitions between these states are non-covalent interactions. They correspond to the enzyme's ability to hold on to the molecule, which is a property of the molecule's overall shape and the type of functional groups present. It reflects the shape of the active site of the enzyme in relation to the atoms that undergo chemistry. In our dipeptide example, it corresponds to the encapsulation of what types of R groups will be tolerated in their positioning relative to other atoms in the molecule. This type of model is generally referred to as a structure-activity relationship, or SAR model. Suppose we have two enzymes, E1 and E2, and we want to know if these two enzymes represent a process or not. For there to be a process involving both enzymes, then one of the outputs of E1 must be an input to E2. For this condition to be met, some molecule P1 must satisfy the constraints of E1's transitions and also satisfy the SAR constraints for binding to E2. If this condition is met, then this reaction sequence of A to B to C involving E1 and E2 could occur. If the product of E1 matches the SAR constraints for substrate binding of E2, but that molecule does not satisfy the mechanistic constraints for conversion, then it is a competitive inhibitor of E2. Genetically, we would express that by saying that the E1 represses E2. If no SAR constraints match, these enzymes are orthogonal to one another and no process is going to occur. If we combine these two enzymes in an aliquot of liquid and feed them substrate A, the overall system now has 9 different species. This combination of molecules can now be referred to as a process. We could define that process by the set of molecular functions, which would be the set of molecular models for E1 and E2, or we could express it as the pathway from A to B to C. When a pathway is expressed in the cell, the interactions that are observed become concrete. Either there is some specific molecule A in the cell that undergoes this pathway, or nothing happens. However, in another cell, a different molecule could satisfy the role of A as long as it satisfies the SAR and mechanistic constraints for the enzymes in the process. Thus, once we define the genetic composition of a cell, the distinction between the set of molecular functions and the pathways observed is irrelevant. They correspond to the same chain of events. However, in synthetic biology, we are often adding foreign genes to a cell, and there is no guarantee that the same cohort of substrates will interact with these enzymes as did in the source organism. For example, other enzymes in the chassis organism may generate other molecules that satisfy some of the constraints for the enzymes. If those molecules satisfy all the constraints, then a side reaction is observed. If they match SAR constraints but not some mechanistic ones, then they could inhibit the pathway. Thus, when we introduce a series of genes that performs a function in one cell to another cell, it is not necessarily the case that the same process results. Additional interactions may be taking place that complicate things. The term device in synthetic biology refers to an isolated subset of genes that together encode a process. A device refers to either the process in its entirety or just the genetically encodable portions such as RNA and protein coding sequences. You can encode a device in DNA by creating a part, which in this case would involve a promoter and two open reading frames. There is a fair amount of debate about the definition of a device in synthetic biology, just as there is a debate about what is a part. The different definitions all amount to different levels of abstraction. For example, does the definition of the device include the RBS or not? And does it correspond to a specific protein sequence or just the specific molecular function of the enzymes involved? The gist of all definitions deals with some process that is encoded in a DNA, but the most useful abstraction for engineering is not yet fully defined. When you transfer a device to another cell, assuming all the enzymes still get expressed and folded and all the native inputs are provided, then the full set of molecular interactions should be preserved and the process will still occur. However, additional reactions will also occur and could have the effect of creating alternate products or interfering with the process encoded by the device. In this discussion, I've spoken specifically about biosynthetic pathways with chemical inputs. However, as drawn and formalized as state machines, there is no requirement that these inputs are small molecules. They could be proteins, RNAs, or DNAs too. In all scenarios, you can model the function of a biomolecule in terms of microstates and non-covalent and covalent transitions between these states. Thus, these principles apply to all types of devices and processes.